Hello, I am Brockton Richards, and I have the privilege of presenting to you the final result of Group 16's research. We all worked very hard and enjoyed learning about this fascinating technology, and we hope that you have as much fun learning about this as we have. Today, I will be covering the topic of organ on a chip, what it is, the pros and cons of organs on a chip, and the difference between animal testing and organ on a chip. First, we will dive headfirst into what exactly this chip is, how it works, why it works, and how it's made. So, what is an organ on a chip? Basically, an organ on a chip is a tiny, clear device about the size of a USB stick that contains real, living human cells. The whole point of it is to mimic how an actual human organ works, but in a small, controlled environment. These chips are usually made from flexible plastic, and inside them are tiny hollow channels that are only a few hundred micrometers wide. That's pretty small, about a millionth of a meter small. These chips are intended as an alternative to our current methods of drug testing, which is primarily animal testing. It is well known that animal testing business has had many ethical problems, but beyond this, these tests were inaccurate, did not reflect properly how the human body would truly react to various drugs and stimuli. Inside the chip, these channels are separated by a thin, porous membrane that's coated with something called extracellular matrix. That's basically the natural glue and support system that surrounds cells inside our bodies. Because the membrane has pores, we can grow two different types of tissue on each side of it. For example, one side can have epithelial cells. These are the cells that line surfaces like our lungs and intestines. And on the other side, we can have endothelial cells, which line our blood vessels. Putting these two layers close together recreates what's called a tissue-tissue interface. That's important because this is where a lot of real organ functions happen, things like gas exchange, nutrient movement, and immune response. And because we can flow fluid through channels, almost like artificial blood flow, we can watch how these cells behave in a much more realistic way than in a regular petri dish. The design has managed to replicate the tissue-tissue interface at a microscopic level, all while also perfectly replicating organ-like functions. This and providing a better, more realistic result than a petri dish. This essentially turns our USB-sized chip into a microscopic punch-out of a specific organ. What's even better is that this technology isn't limited to just one organ. In fact, it is capable of recreating our brain, gut, lung, kidney, liver, and even our blood vessels. Scientists are working hard to broaden the capabilities even further, and we have much more applications to this technology to look forward to. This technology has amazing capabilities. We're able to better recreate the interactions certain drugs have with human physiology without having to deal with all the struggles that can come with animal testing. Not only this, organ on a chip also has more advanced, real-time monitoring of how each organ tissue will react. This essentially means that here we have a more morally acceptable, more accurate, and faster recreation of an industry that has been notoriously slow for years. It is plain to see that this technology has the capacity to change food and drug business permanently for the better. While the organ on a chip is great, like everything else, it has its fair share of drawbacks. These chips cannot recreate the full, intricate, complex organ in every single one of its interactions. These chips tend to be expensive and very complex, making the production process a little tricky, meaning these chips are also fully not standardized yet. However, as more and more research and development goes into this technology, we get closer and closer to overcoming each and every one of these hurdles. So, in simple terms, organ on a chip works because it combines real human cells, the right physical structure, and right conditions like flow, nutrients, and mechanical forces to recreate small versions of human organs. This gives researchers a realistic way to study human biology, test drugs, and understand diseases without needing to go straight to animal testing. I hope we were able to shed a little more light on this wonderful technology for you. Thank you very much for watching. We have attached a relevant quiz in the description below. We hope you get the chance to check it out. Thank you very much.